Measure 12 ml of 3 mol of hydrochloric acid and then transfer it into a 100 ml volumetric glass. Transfer 25 ml of amine solution by using a pipette into 3 different beakers. Measure 75 ml of 0.1 mol hydrochloric acid and pour into the beaker containing amine solution to dilute it.
To each beaker, add 5 drops of methyl indicator solution using a dropper bottle and stir it with a glass straw. While stirring it, one of the group members will weigh ammonium oxalate using a graduated cylinder. Add approximately 25 ml of the ammonium oxalate solution to each beaker and stir it with a glass straw. Next, weigh the filter paper for the filtrations and the solid urea, about 50 grams, using analytical balance. After that, label each beaker and add a solid urea to each of the beaker and stir it with a glass rod. Cover each beaker with a watch glass to prevent splattering and minimize evaporation. Place the beaker on a hot plate and boil the solution gently for about 30 minutes until the indicator turns yellow. The watch glass should remain on. Now we will separate our solid precipitate from the liquid. Pour the dilute water onto the filter paper and use the glass rod to remove the bubbles from the edges of the filter paper. Filter each hot solution through the funnel using suctions and use a rubber policeman to dislodge and help transfer any remaining solid to the funnel with 10 mm portion of ice cold water to purify it. After putting the filter paper on the petri dish, place the petri dish into the oven for 1 hour with the temperature of 105 degrees Celsius.
Make sure to properly dispose of chemical waste into the bottle prepared by the lab assistant. After drying for 1 hour, record the mass for each precipitate form.
This is the raw data we got from all trials. From the experiment, we need to find the weight percent values, the mean, and the standard deviation using manual and Microsoft Excel. The trial masses are 0.27 grams, 0.22 grams, and 0.18 grams respectively. The total molar mass for the precipitate is 146.116 grams per mole. The mass fraction of calcium is 0.274 based on the formula. The weight percent for all 3 trials are 14.8%, 12.06%, and 9.86% respectively. The mean is the average, which is 12.24%. Based on the calculation, the standard deviation using the formula is 2.48%, and the relative standard deviation, which is standard deviation over mean times 100%, is 20.3%. Based on the Microsoft Excel, we got the same amount as in the manual calculation, whereas B5 is the mean, B6 is the standard deviation, and B7 is the relative standard deviation. The RSD of 20.2% is very high. This indicates poor precision in the results. The values from all 3 trials are spread out widely around the mean. A low RSD, often below 2 or 5%, is desirable as it indicates high precision and low variability. And from our experiment, an RSD of 20% suggests sources of errors, such as loss of precipitate, incomplete precipitation, inconsistent drying, impurities in the precipitation, and technique errors. In order to improve our results, we should carefully review our technique in steps of precipitation, filtration, and washing for accuracy in the results. The experiment was successful as we found the average calcium content to be 12.24%. However, the results were not precise as the RSD was very high at 20.3%, which indicates all 3 measurements were far apart. This suggests errors occurring during the lab, such as filtering and not drying the sample consistently. Hence, the calculated value of 12.24% is not really reliable.